Before meiosis begins, the cell must grow and the DNA must replicate. This occurs in the process interphase. In gap 1, the first stage of interphase, normal metabolic functions occur in the cell, as well as cell growth. In the second phase, synthesis, the DNA of the cell replicates, producing 2 copies of each chromosome. The cell will still have 46 chromosomes, but each chromosome is double DNA, so there is 92 sister chromatids. However, the DNA is in the form of chromatin until the first phases of meiosis. In the final stage of interphase, gap 2, the cell continues to prepare for meiosis and cell division by continuing its growth. The centrosome, containing centrioles, also duplicate in this stage. The cell is ready to undergo meiosis. There are 8 major steps in meiosis: prophase 1 and 2, metaphase 1 and 2, anaphase 1 and 2, and telophase/cytokinesis 1 and 2. In early prophase 1, the DNA coils tightly such that chromosomes become distinct. As prophase 1 continues, chromosomes condense further and the nucleolus disappears. The two centrosomes begin to move away from each other and migrate to the opposite poles in the cell, as a spindle starts to form between them. Then, in the process of synapsis, homologs, double-stranded chromosomes find each other, forming a 4-chromatid structure, or a tetrad. During synapsis, crossing over occurs. This is where segments of non-sister chromatids in the same tetrad break and reattach to the other chromatid. This process produces genetic diversity in the offspring, such that no one chromatid is identical to another. The spindle fibers attach on the centromere and pull the chromosomes apart. Anaphase has now begun. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. During cytokinesis and telophase 1, a cleavage row forms in the equator of the large cells and separates the cell evenly as the nuclear membranes form as well. There are now 2 daughter cells, each with the haploid number of chromosomes that are double-stranded. The process starts again in prophase 2 with the nuclear membrane dissolving. Once again, the spindle fibers start to reach out to grab the aligned chromosomes in metaphase 2. Next, they pull the chromosomes apart to the opposite poles in anaphase 2. Lastly, a cleavage furrow forms along the equator of the 2 daughter cells, splitting them into 4 finished daughter cells. The end result of meiosis is different depending on if it is a sperm or egg cell. In a sperm cell, the process is called spermatogenesis, and oogenesis is if the end result is an egg cell. In spermatogenesis, the diploid cells are called spermatogonium and results in 4 spermatids, the haploid cells that mature into sperm cells after all divisions. Spermatogenesis occurs in the testes, and men produce about 250 million sperm per day. If meiosis is occurring in the ovaries, the process of oogenesis. In oogenesis, the 2 meiotic divisions produce 3 polar bodies that die in 1 ovum or egg. This is the result because the egg cell is much bigger than the 3 polar bodies, so the polar bodies die because they cannot survive the unequal division of cytoplasm. However, meiosis II does not occur until fertilization. An egg cell differs from a sperm cell such that an immature egg is called an oocyte, diploid, which then matures into an ovum or egg every 28 days after puberty. Also, oogenesis begins before a female is born, while spermatogenesis occurs daily for men. This means that women have a set amount of egg cells, about 400,000 by puberty, while men are continuously making sperm.